Hello, my name is Christen. I am an assistant professor in systems biology working at ETH Zurich in Switzerland. I was in middle school and probably enjoyed one of my first classes in biology. I was fascinated about all different types of living beings. And as a small child, I dreamed to become an inventor that designs new types of flowers. Did this wish come true? No, I have not yet come up with a new type of flowers like a blue sunflower. However, as a bioengineer, I am still very interested and fascinated by our living world and doing research on the molecular blueprint of life. Our research breaks the wall from reading genomes to start writing genomes. Through chemical DNA synthesis, we can start write genomes anew for applications in medicine, agriculture, or in industry. For instance, we can start to develop microbes that keep us healthy and cure disease. Such tailor-made microbes have great potential to address global challenges using nature as our inspiration. Genomes from even the simplest organisms, such as microbes, are huge DNA molecules comprised of millions of DNA letters. Synthesizing such a genome from scratch is a daunting feat. It's very time-consuming and costly. So, what is our solution? Our research group develops computer algorithms that allow us to write genomes anew. These algorithms identify all the molecular parts needed and then design a new computer-generated sequence that is very easy to synthesize from a chemical perspective. Together with my team, we have built the first computer-generated genome of a bacterium. We took a freshwater bacterium as a starting point to physically produce a new genome. Although a corresponding living cell does not yet exist, we have introduced portions of that genome into bacterial cells and found that most genes work as intended. So this tells us that biological systems are so simple that we can define them on the computer according to our needs and then build them. Our research will help to tackle global challenges. For instance, we can start building microbes to build better vaccines and come up with new therapies, reduce our reliance on fossil fuels, or increase the sustainability of agriculture. For instance, we can start building microbes that protect and fertilize our crop plants, thereby reducing the need for chemical fertilizers. There are myriads of microbes living on our planet, and in the detail we don't know how they are working at the molecular level. So synthesizing genomes allows us to study these organisms. Which part of your research keeps you awake during night? With recent technological advances, I'm convinced that soon we will see designer microbes become reality. So we need to reanalyze and expand the national and international framework of regulations considering the manufacturing and trade of synthetic DNA. It is up to us to ensure that this technology is used for the greatest possible benefit for society. When my 4-year-old daughter asked me, "What are you doing the entire day at work?" I took her to the lab and showed her the world of microbes through a microscope. Now she keeps me asking, what are these microbes doing? Do they sleep? What type of food they eat? And what are they doing?